folks, welcome back. We're going to look at genetics today. So we've already looked at DNA and how that DNA can be used to make proteins. But when we make those proteins, how are they expressed? All right, how do we know what traits we're going to get? And how do those proteins express certain traits? So when we look at genetics, we're really looking at how does the DNA that we have in our bodies create traits that are exhibited in ourselves. All right, so how does the DNA get to becoming to show different traits? And we're going to look at that today. So hopefully you'll be able to understand and explain some of the principles of genetics, analyze some key terms, because terminology is very important here. You're going to hear a lot of terms that you may not necessarily be familiar with, but by knowing them, you're going to be able to do a lot of the problems that are present in this particular genetics unit. Also, you should be able to predict the products of a simple monohybrid cross. All right? So, the first thing we need to look at is how has genetics changed? And to understand how genetics has changed, we need to look at how genetics got started. In 1866, a man by the name of Gregor Mendel studied the inheritance of certain traits in pea plants. As he explored this, he thought about different organisms that he could use and found that pea plants would actually be the best because they were easy to use and he could control the breeding. Whereas in animals, you can't necessarily control the breeding quite as well as you would if you were to use other organisms. All right, so pea plants were by far the easiest to use because they were easy to look at, easy to determine traits, and easy to reproduce. What Mendel noticed is that he bred pure yellow peas with pure green peas and found that he ended up with all yellow peas. And he found this very bizarre, that yellow peas and green peas, that you get all yellow peas. So what he did is that he bred all of the yellow peas from the offspring and found that he got a 3-to-1 ratio, so that for every 4 plants that were produced from breeding the 2 yellow pea plants together, he got a 3-to-1 ratio of yellow peas to green peas. So 75%, 3 out of the 4 were yellow, and 1 out of the 4 were green. He determined that genes must occur in pairs. One gene color masks another. So these genes that we get in our bodies must be in pairs, one coming from mom and one coming from dad. And that pairing, that combination of pairs determines what you see. It determines what traits you have as an individual. Now, we know a lot more about it now than he did back then, but this is an amazing discovery. He was able to determine that genes have to occur in pairs. And as a result of this, certain traits can actually be masked by other traits if given a proper scenario. So green and yellow are alleles of a gene. It's just different forms, just like anything else, whether it be eye color is an allele, hair color is an allele, height is an allele, the ability to roll your tongue is an allele, all right? What he noticed is that the yellow allele, if an organism had both one yellow and one green, remember, each organism has 2 genes for this. So if he has one yellow allele and one green, that the yellow masked the green. It wasn't some combination. It was the fact that the yellow gene masked the green gene. That means that yellow is a dominant trait and green is a recessive trait. So if I have an organism that has one of each, it's going to be yellow. If I have an organism that has 2 yellow genes, it's going to be yellow. The only way it could be green is if it has 2 green alleles. Remember, if it has one yellow and one green, it's still going to be yellow because yellow is dominant to green. Okay? Dominant traits mask recessive. So the big letter, when we abbreviate this or write this out, the big letter is dominant. So for example, if we were to use the colors here, big Y would represent yellow, and green would be represented by the little y. All right? So dominant is represented by big letters and recessive by little. Remember that 2 alleles are present for every single trait. This is what Mendel discovered, is that there has to be 2 alleles for each trait. Otherwise, how could we get green plants from yellow plants? The term we use to describe organisms that have 2 of the same allele is homozygous, right? I mean, homo meaning the same. So when we have 2 different traits, big Y, if we use an example as before, is homozygous dominant. It has 2 of the same allele that are dominant. So big Y is homozygous dominant. Little y is homozygous recessive. Heterozygous, hetero meaning different, would be one of each allele, big Y, little y. So if you think about the trait we last talked about where big Y is dominant to little y, we can see that big Y will be yellow, right? They'll have yellow pea pods. Little y will be green because it has two green alleles. And big Y, little y will be yellow because the dominant Y masks the recessive. So make sure you know these terms because they appear often in our genetics problems. Homozygous dominant and recessive and heterozygous. A couple other terms that we use, genotype and phenotype. The genotype is the specific combination of alleles. That means, is it big Y? Is it big Y? Is it little y? All right? That's our genotype. It is the specific combination of alleles. Genotype. Easiest way for me to remember that. Phenotype is what the organism looks like. So if we looked at the pea pods, they're either yellow or green. So that's one way we need to look at this. There are two ways to analyze each problem. We need to look for genotype and phenotype. What are the combinations of alleles and what do they look like? A couple of laws to understand here. And then that helps us make what's called a Punnett square, and Punnett squares are going to help us analyze different genetics problems. The law of segregation states that the two alleles for each trait will separate during meiosis. We're going to receive one allele from each parent. All right, and these alleles unite during fertilization. So if you have a certain trait, you have two alleles for that. But you're only going to pass one of those alleles on to your offspring. The other allele is going to come from the other parent. After fertilization, that zygote then has two alleles. Remember, we talked about this with meiosis, right? You start with 46 chromosomes. You pass half of those on to your offspring. The other half come from the other parent, and you end up with 46 again. It's the same thing here. It's just saying that the law of segregation says that two alleles for each trait will separate during meiosis, and that it's completely random on which trait you will receive from your parent. We use Punnett squares to predict the possible genotypes and phenotypes of our offspring. And to do this, we need to draw a square, and then we need to separate the alleles that are present in both of our parents, cross them, and then analyze those boxes for genotypes and phenotypes. It takes some practice, but it's pretty simple once you get the hang of it. You just need to make sure to read the problems carefully and go through each one step by step. So for example, let's take a look at the Punnett square problem that is present to us. Tongue rolling is a dominant trait and non-tongue rolling is a recessive trait. Cross two individuals who are heterozygous for that trait. Determine genotypes and phenotypes. Now realize this is a lot of information that is given in one particular problem, but let's break it down, all right? Let's break this down really, really easily. Tongue rolling is a dominant trait. Non-tongue rolling is a recessive trait, all right? First information we have there. So what we should probably do is go ahead and write out some information. Tongue rolling is dominant and non-tongue rolling is recessive. So let's assign them some letters. All right. Big R is rolling and little r non-rolling. All right. Now, let's take a look and see what else we have here. We've got two individuals who are heterozygous. So we got two that are heterozygous. If you remember, heterozygous means that you have one dominant allele and one recessive allele. So we're going to cross two individuals that are big R, little r, and big R, little r. Okay? So let's go through that step by step. Now remember, when you draw out your Punnett square, you need 4 boxes. Just like that. And we're going to place each allele into each box. So big R, little r, we're going to place one letter here and one letter here. Now it doesn't matter which way you put it. So we're going to put big R there and little r there. Again, we're going to take each allele and put it into the top boxes. And then what we're going to do is we're going to cross them and see what type of offspring we get from this. So here we're going to have big R, big R, big R, little r, big R, little r, and little r, little r. Okay? Now, from this, we need to determine our genotypes and phenotypes. So let's take a look here at our genotypes. Okay? The first question we need to ask, how many of the offspring are homozygous dominant? That is, how many have two dominant alleles? Well, this one has two dominant alleles. So we have one, big R, right? How many then are heterozygous? Two. Two, big R, little r. And then that leaves one that is homozygous recessive, little r. Okay? When we look at phenotype, that is what they look like. So how many of these individuals are going to express the dominant trait? This one will because it has two dominant alleles, right? And these two will because the dominant masks the recessive, right? So we're going to have three individuals that can roll their tongue and one individual who cannot roll their tongue. All right? That is the information that you need to be able to determine from a Punnett square. All right? So we need to be able to break down the problem, read what it says, and then analyze the data to determine what offspring we're going to produce. Let's go through another practice problem to get a better understanding. Brown hair is dominant to blonde hair. Cross a male who is heterozygous for brown hair with a blonde-haired woman. So, let's go through this together. Brown is dominant to blonde. Let's figure that out first. Let's assign traits. We're going to use B's here. So, big B is brown. And little b is blonde. All right, good start. We're looking for a man who is heterozygous and a woman who has blonde hair. Well, we all know that heterozygous is one dominant and one recessive trait. So that's going to be big B, little b. And we need to cross that with a woman who has blonde hair. Well, what is the only possible way that a woman could have blonde hair? She has to be little b, little b. Any other combination would result in brown hair. So we have to cross these two individuals. The rest is exactly the same as before. We draw our Punnett square. We fill in our alleles. And we cross them. Now, let's take a look and analyze our data. Let's do genotype. How many of the individuals are homozygous dominant? None, right? We have 0 individuals that are big B, big B. How many are heterozygous? We have 2 that are big B, little b. And how many are homozygous recessive? 2, little b, little b. Okay? So 0 that are dominant, 2 that are heterozygous, and 2 that are homozygous recessive. Phenotype is last. How many will be brown hair? Hopefully you answered 2, because the individuals that are heterozygous will have brown hair. And then there will be 2 that are blonde. Because those 2, you have 2 individuals who are homozygous recessive. Okay? So do you see how to go through and analyze this? We need to look at both phenotype, which is what they will look like, and genotype. All right? Let's do one more. This problem is a little bit trickier because it's asking you to find some other, it's not asking you for offspring this time. It gives you offspring, but you need to figure out what the unknown parent is. So let's analyze this. Tall is dominant to short, so let's go ahead and label that. We're just going to use big T for tall and little t for short. A plant that is short breeds with an unknown genotype to create offspring that are all tall. So we know we've got a short plant. So we know that one of our parents must be little t, little t. But the other parent is unknown. What we do know is that all of our offspring are tall. So all of our offspring have to be one of two genotypes, right? They're either big T, little t, or big T, big T. What can we put in the box here that would create offspring that are big T, little t, or little t, or I'm sorry, big T, little t, or big T, big T? If you think about it, the only way that we can get any of these is if the parent is big T, right? All of the offspring have to be tall. That means we can have no short offspring. So if we go through and do the Punnett squares, we'll find that the only way that we can get all of our offspring to be tall is if we have the other parent be homozygous dominant. Okay. Anything else that we put in there, if we put little t, that's going to give us all recessive. That's going to give us all short plants. We can't have that. Even if we did big T, little t, we're going to get some tall plants, but we're still going to get some short plants. Remember that all of the offspring are tall. Okay? So as a result, we have to have a parent that's going to give us all tall offspring. And the only one that does this is this one right here. Okay? So hopefully you understand this a little bit better. We're going to spend some time getting some practice through these, and I'm going to go some more of these examples with you a little bit later on. Hopefully now you're able to understand the principles of genetics, analyze some key genetics terms, and predict the products of simple monohybrid crosses like what we've just been doing. Get some practice with this. This is really important for your test. I wish you nothing but the best of luck. Be sure to ask me questions if you have any, and we'll go through some details a little bit later on. Have a great day, guys. We'll talk to you later.